All right, we're live and welcome everyone. And thank you to Dr. Laszlo G. Boros for joining us today. We're going to be talking about deuterium and deuterium depletion. And we're talking about keto, carnivore, and many other things. And this is going to be for replay. And really grateful to have Dr. Boros here, Doctor of Medicine degree at Albert Szent Gorgi School of Medicine in Hungary, and retired professor from UCLA in Los Angeles in pediatrics, endocrinology, and metabolism. And I'm from Los Angeles, so we've got some things in common. But I'm going to let him tell us a little bit more about himself and how he got into this world of deuterium and depletion and help us understand it better. So Dr. Boros, thank you and welcome.
Dr. Kiltz, first of all, thank you very much for inviting me to participate in your conversations, and it is an honor, and I would like to thank you for all the work that you're doing regarding nutrition and ketosis and deuterium depletion for that matter, because they go hand in hand. And the medical, I'm just like yourself, I'm a medical doctor. I was graduating in 1987 from the Harbor-UCLA Medical School. He was given the Nobel Prize in 1937 for the discovery of vitamin C, and also the Sanjari-Krebs cycle. He clarified a few reactions. Fumarate hydratase was one of them, which is part of the human Ferrari, the engine for that matter. I find your selection of topic very appropriate, simply because we are Ferraris in many sense, and also our engine is a high performance, maybe even like a Formula One race car engine, in that sense that because these mitochondria, which are now in the focus of many conversations, and the mitochondrial membranes in these proteins, they actually deliver biological power, and they work like nanoturbines or nanomotors for that matter, to generate biological energy, which is ATP, and that has relation with all kind of physiological and biochemical processes, including fertility, and many common population diseases, obesity, cancer, and so on. And so I went to Germany first. I spent about 2 years at the Essen University working on pancreatic cancer and chronic pancreatitis. Then I joined the historic laboratory of Dr. Ellison and Zollinger at the Ohio State University. And I actually did know Dr. Zollinger in person because he was still coming in for eminent conferences. And then in 1998, I got my faculty position at the University of California, Los Angeles, UCLA, Department of Pediatrics, and I was an active teacher and researcher up until July 2021, when I retired from UCLA, and I started teaching deuterinomics or deuterium biochemistry or medical applications of deuterium sciences at the Vrije University in Amsterdam. And now we are also teaching the same or similar topics at the Medical School of Amsterdam. And we are expanding the course, deuterinomics course. Deuterinomics is practically the name of the science that is entering a credit course scenario, which is transferable doctoral credit at medical schools and doctoral schools. So, in fact, the first lecture is given by Dr. Judith Kleeman, who works at Berkeley. So her research was for a long time was covering deuterium kinetic isotope effects. And this is practically the medical translational application of this very fundamental basic research. And so now I also do editorial and review work for major medical journals, including Scientific Reports, which is a Nature publication, and Medicine, and also for Metabolomics, which is the prime scientific journal for the field of metabolism or metabolic research. And I did do a little bit of clinical work when we did clinical trials at Harbor-UCLA, which you also went to train in medicine, in fertility research. And so that I was pursuing and I was actively involved in research there for about 20 years. So in a nutshell, this is my background.
So deuterium, maybe you can just, for so many people that probably don't know much about the science and the metabolism, just a brief discussion on what is deuterium and why is it important?
Yes, indeed. And this is the fundamental question, what deuterium is and how and why is it so important in biological systems. Our body is mostly made up of hydrogen, and hydrogen is the first element of the periodic table, and hydrogen has a role in chemical bonds. We call them hydrogen bonds. Hydrogen has a very important role as a structural molecule or binding biomolecules together, and it has a role in rotating enzymes or rotating proteins by its positive charge and generating energy for our body. We obtain hydrogen from food, which is when you see a H in any molecule that you look at, it's a hydrogen that is transferred in mitochondria to the intermembrane space, and as hydrogen comes back into the matrix, it delivers practically a rotating type of force on a nanomotor, which is the engine of the Ferrari, of the human Ferrari. We call them complex 5 or ATP synthase. These proteins, these spinning proteins, these nanomotors are sitting in the intermembrane of the mitochondria, and these are designed to use protons or hydrogen ions with a positive charge to rotate at an optimal 40,000 rotations per minute in some of the cellular phenotypes that we have studied, but they rotate anywhere from 5 to 90,000 based on what type of species or what type of cell or even bacteria we are investigating or looking at. Now, deuterium comes into the picture simply because deuterium is the brother or the heavier brother or the heavier sibling of proton or hydrogen in nature. The proton has a proton and an electron, that's hydrogen, and deuterium has a proton and a neutron in the nucleus and an electron. And that means, and that makes it twice as heavy, and as far as the nucleus is concerned, twice as large. That means it is really not the type of isotope that fits into these nanomotors. It is really not an isotope that is easy to detach from chemical bonds, and it's really not an isotope that is easy to move around in our system, yet it is part of, because of his younger brother, hydrogen, it's part of every vital reaction, chemical reaction, or rotating nanomotors in our body. That means it's more like a sibling that is twice as heavy, twice as large, and being able to participate in similar biochemical reactions or the same biochemical, but practically destroys everything in our mitochondria. It's like it behaves like a bull in a china store. As far as mitochondria are concerned. Now, actually, deuterium, because of its chemical properties, it is participating in connective tissue formation, bone formation, for that matter. In collagen, which is a protein that is more of a structural, not a rotating protein, but a structural protein, deuterium, because of its heavy or stronger bond to other molecules or other atoms, it actually makes those bonds 8 to 10 or 15 times stronger than hydrogen would. And for that matter, some bone structures and bone collagen structures are prone to collect deuterium in our system and spare the nanomotors of our mitochondria as far as rotations or rotating force or power is concerned. It's pretty much like looking at your Ferrari, where you have an engine that has to run on clean fuel, hydrocarbons, or gasoline, and the engine needs, or the car needs, structural parts like spring absorbers, it needs a fuselage or a body, a hood, and so on. And those are actually made up with chemicals with different deuterium content. And for that matter, our body has a very sophisticated system to differentiate between hydrogen and deuterium, to locate them or send them to specific parts of our rotating proteins and stable proteins or structural proteins. And in the meantime, if there's too much deuterium in the system, that means your body or your cells is not able to easily separate, discriminate, and fractionate them. They end up at the wrong place at the wrong time. And that's the mitochondria. When deuterium gets in there, that's the wrong place for deuterium. And if your food is overloaded or has too much deuterium in it, then your cells are not able to kind of differentiate between deuterium and hydrogen, and they end up in mitochondria, and they break these nanomotors. And once these nanomotors are broken down, then your TCA cycle, your Krebs synuclein cycle, your oxidative phosphorylation, your energy production in your cells, they become defective. And you start looking at a whole host of metabolic and cellular proliferative diseases, for example, cancer, diabetes, obesity, degenerative diseases, infertility, and you just name it practically. We did find a role of hydrogen deuterium ratios, or the proportions of how much hydrogen there is in your food and in your body fluids and your body components, in your molecules, how much deuterium and how much protons or hydrogen there are, that will fundamentally determine how your body functions. And this is the foundation, this is the bottom line, this is the substance of why deuterium discrimination, deuterium fractionation, or separation of deuterium from hydrogen is so important in our system, and why food is so important as far as how much deuterium your food contains. And there are two major components here that we need to consider. One is that what food you eat, how many times you eat, how much food you eat in general, how much you drink and what you drink, and what's the source of all of these. So these are the two main components that we need to consider when we look at deuterium or this heavy hydrogen chemistry.
That is all amazing, and breaking that apart and simplifying it. You've done such an amazing job. I'm just wondering if we can look at it like premium fuel and regular fuel, sort of supercharged fuels. You go or to jet fuel and leaded standard fuel for most cars. If that's a good way. So when we say hydrogen, we're talking about a lighter form, and then deuterium is a heavier form essentially, but we're really getting to the atomic levels to understand what's going on where it really matters, probably more than anything in this. Would that be the simple way of sort of saying that deuterium is a form of hydrogen that is not as well processed in our bodies in order to make the energy, or it's more like leaded fuel, where the standard hydrogen is more like premium fuel. It burns cleaner and better, and the engine in the Ferrari just functions better.
That's very true. That's a very good description of the situation. A high quality gasoline, for example, a high octane gasoline. Actually, octane is a 8-carbon fatty acid oil in your gasoline, and that burns the easiest, that burns the fast. That's the ketogenic substrate or the source that burns in your engine. And higher this short chain oil or fatty acid is in your gasoline, higher quality is your gasoline, because it only contains carbons and hydrogens. If there is lead in the gasoline, for example, it's just not as efficient by burning it, then your engine is going to have a wear because of this particular phenomenon. And besides that, it seems like when deuterium gets into your engine, it's more like a sand or more like a piece of your spring absorber got into the gasoline somehow. So your system is not operating, your engine is not operating with the optimal fuel. Go ahead.
Well, so you mentioned nanomotors, which are essential protein motors that move and turn in order to keep our
ATP synthesis.
ATP synthesis moving into making ATP, and
That's
the
right.
the gradients of the molecules of the electrons and protons or hydrogens in the right spot. And so, that's something I think most people don't understand, that we have moving parts at the cellular level, and the fuel that you feed it is very critical.
Yeah, you captured it right. It's what we call the submolecular medical sciences, and this is where it comes down to. There are rotating parts like hairdryer parts in your mitochondria, in your cells, and they rotate by the force of a proton gradient between the two sides of the mitochondrial matrix membrane. And because of this proton gradient, these nanomotors can form or can turn at certain speed. And the reason why this gradient is maintained, because once the proton falls into the mitochondrial matrix, it is united with oxygen. This is why we breathe in oxygen, and this is why through your carburetor you need oxygen in your engine, because this oxygen with the hydrogen or the proton that just power these rotating nanomotors will form water. This is what we call metabolic water, or matrix water for that matter. And we produce large amounts of protons, or we rotate large amounts of protons per nanomotors. Each nanomotor per second, they uh, drop in, or they move about 1,500 protons from the outer membrane into the matrix where water is formed with the help of oxygen. So practically how the system works is like a car engine or Ferrari engine. You need oxygen through the carburetor, and you need gasoline for the hydrogen that is united in the piston, above the piston in your cylinders, where there's a spark plug that activates oxygen, activates gasoline for that matter, and the oxygen and hydrogen unites very quickly and delivers a explosion or explosive force. This is how fast oxygen reacts with hydrogen. And this is what happens in mitochondria also. So your description of a nanomotor or the Ferrari motor is very appropriate, simply because the same chemical reactions happen. And when you actually park your car in front of the garage, while you're opening the garage, there's water dropping out from the exhaust pipe. That's your metabolic water that is produced in your engine. And in the engine, you also rotate a camshaft, which is the same in the nanomotor. There's a rotating part, or F1 part in the nanomotor itself. It has a static part and it has a rotating part. And this rotating part is very, very sensitive to the amount of deuterium in your food, just like your gasoline in your car is very sensitive to how much lead there is in there, or how much sand or sand crystals there are. That's why you have filters, gasoline filters in the gas tank, and you have gasoline filters in the carburetor, and you have sedimenters where you actually more like your gasoline is decanted just to make sure it's clean. And this is why you have to use oil filters as well, to get rid of every kind of mechanical part, little pieces of metal that can get into your pistons in your cylinders that can damage your engine. So the biology of internal combustion engines is practically the nanomotors in the mitochondrial matrix, and the water formation is practically how the most efficient way is accomplished in delivering energy and ATP, biological energy, for your cells. And you need this energy for many reasons. You need to keep ion gradients, so your nerve cells, neurocytes, can run action potentials. You need it for fertility, because sperm are powered by nanomotors, very much the same way as in any other cell. And they all work in the same scenario. They burn fuel, which is mostly fatty acids and ketone bodies that carry only carbons and hydrogen. Carbon is disposed in the form of carbon dioxide from the substrate itself, and oxygen from air is used to make metabolic water, which is deuterium-depleted metabolic water, as far as the mitochondrial matrix is concerned. And we actually move a high volume or high mass of protons each day. For example, an average individual would move protons equivalent of 2,000 gallons of water a day. And this is how much blood you circulate through your heart. If you calculate the ejection fraction, which is about 90 milliliters, the heartbeats, you come up with the same number as how many times your blood is circulating through your heart. It's about the same amount. And in the meantime, oxygen is connecting the outside world with your mitochondrial matrix to produce metabolic water. And for that matter, this is a sophisticated, beautifully designed. It's in the Bible in many sense, this type of operations described and referred to. For example, the fifth book of Moses is called Deuteronomy, which carries the same principle. And all the nutritional recommendations of in the Old Testament and all the reference to water and the spirit hovering over the water, which is also due to depleting process. It seems that this knowledge that we are talking about, the knowledge that we are covering here, it goes back in human history, like very early times. I would say as early as the caveman came to life, or the caveman was adapting to the environment, for that matter. And it seems that all these elements and all these parts of the human Ferrari, or the human body, or the cells, are somehow referred to in old text as principles, or more like basic fundamental information that we can utilize nowadays to understand human history and biology and anthropology in a more comprehensive way.
So what seems to happen in our modern world as a researcher and a physician that's practiced in the pediatric world, and it sounds like in the cancer world, we're suffering more cancer, more diseases, more metabolic diseases, diabetes, obesity is on the rise all over the world. And bringing this all together, we talk about mostly people are eating too much, or they're eating too much of a plant-based diet potentially, and it sounds like we're getting more deuterium damage, which ultimately damages the mitochondria, and I think, as Otto Warburg and others have sort of talked about, which ultimately is the death of the cell. And if you have enough cells that die, the whole organism dies. So how can we sort of put that in understanding of what's the problem? Is it we're eating wrong, or is it deuterium is just on the rise in general in the world, and due to climate and environment? How do we help people sort of figure out the best way to reduce their deuterium exposure and find healthy living so they can live a reproductive life?
This is a very fundamental question, and Otto Warburg did perform some really insightful experiments, and he showed that practically in tumor cells, these nanomotors are broke. And the way he did this is that he implanted tumors into muscles in one leg of the animal, and he was measuring the output of the tissues, the muscle and the tumor together, compared to the muscle, as far as what appears in the blood, provided that the incoming blood carried the same components, glucose and all the nutrients in the same amount, but the muscle that had tumors in it, they produce mostly lactic acid. And this is what happened, and this is what he said. Dr. Warburg, he said only one thing, and that was a genius in that sense, and it was practically the most important metabolic hallmark of cancer cells, is that the fate of glucose metabolism is to produce lactic acid in tumor cells. And to maintain sufficient energy production and proliferation, tumor cells have to use a lot more glucose than normal cells. These were the basic observations of Otto Warburg. And we know that this is true of all tumor cells, or all cells that have broken nanomotors and produce lactic acid. And this is true of adipocytes and defective cells. that lead to these disease conditions that you have mentioned, which are now epidemics practically. And one of the main source of deuterium is, as you mentioned, is food, especially processed food, high fructose food, and so on. And plants, they try to protect themselves. They don't want to be eaten. The fruits are to be eaten by animals that to spread the seeds, for that matter. So it's functional foods. They are functional foods, fruits practically, because they serve the trees to propagate themselves, and they kind of wrap them in the sugary fruit, so it's very appealing to the animals, and so they eat them at once, and then they spread the seeds. But the plant, the green part of the plant, is protecting itself with many different ways. For example, green gas is loaded with cellulose. You have to have a certain type of stomach anatomy, like the cows do, to be able to digest that. And some other plants, they produce toxins. Just you look at some of the mushrooms, produce very high and very potent toxins and poisons, and they also load deuterium simply because they cannot oxidize fat or fatty acids efficiently. So that's why their cellular structures are more compact. They produce chemicals that we cannot, meat eaters or carnivores, we cannot digest efficiently. And I believe, as many believe nowadays, that deuterium load in processed food, in animal food or animal source food that were fed with a agricultural type of fake nutrients. For example, I don't know if you knew that actually they feed cows with corns and leftover candies and protein supplements from jellyfish, dried up jelly. So the cows would not, we actually just published a very big paper in Metabolomics which described these processes. So the animal products that you buy at a, I don't want to say names, but farm-based, farm-raised animals, and this is peer-reviewed medical literature. They are sick themselves, the animals that they offer, and their products, meat products and dairy products. And for that matter, it seems that the safest type of eating and the safest way of remain fertile and healthy without these epidemic diseases like diabetes, obesity, and cancer and degenerative diseases, your best bet is practically to become a carnivore, meaning that to eat meat products, especially fatty meat, that is low in deuterium, and protect and preserve your nanomotors as long as they are necessary to operate in your system, as long as you take in oxygen and food to produce metabolic water, or for that matter, your soul. As you live, as long as you can produce this water. Once you are not able to produce this metabolic water, then that's the state of being dead. And we need to look at this also in a very, I would say, clinically, translationally designed manner to have patients who have various medical conditions. And deuterium depletion through food and through water is very critical to preserve health. And this is what deuteronomics at the submolecular level, as you indicated, would accomplish and would explain and would help physicians and doctors and care providers to make the right decision of how much they eat, what they eat, how many times they eat, and what they drink, because those are all very critical to protect these Ferrari motors that run our system. These are, as you described, rotating proteins that need protection from deuterium. And the best way to do this is through pasture-raised natural fat source, through a carnivore diet, which we are designed to consume and burn in the most efficient way as the most appropriate fuel for our system, high octane pure gasoline, synthetic gasoline, compared to the lead contaminated gasoline that was used in the past and caused a lot of engine damage, obviously, and pollution.
Have you gone mostly carnivore to this the keto carnivore lifestyle?
Yeah, so everybody has to keep in mind that we wake up every morning in the state of ketosis. That's our body. That's what our body is trying to kind of start out in every morning when you wake up. If you measure your beta hydroxybutyrate acid levels, then you can see that those are increased compared to like after dinner, depending on what you ate. And also, this is how every baby is born. They are born in ketosis. That's what every mother after conception are trying to stay with when they are nauseated, when they are thrown up, when they cannot consume just what they see. They cannot really eat as much as they want, simply because your body is trying to keep you and your egg that is that has been fertilized low of deuterium for preventing chromosomal damage and to develop chromosomal abnormalities. And these are all lined up in the same kind of serving the same scenario, and that is how do you preserve ketosis, so running your system on ketone bodies, which are short chain fatty acids or hydrocarbons for that matter, and with a optimal concentration of carbohydrates, glucose, that is produced by your liver. We don't have to consume carbohydrates to produce sugars, because our liver, through hepatic glucose production, can produce glucose and can glycosylate as many proteins as necessary for our epidermal or endothelial cells or in your gut. You can have a microbiome that processes various substrates for epithelial cells and bacteria that help you in maintaining an optimal deuterium content in your body and filter out deuterium for that matter. Because bacteria and yeast, they like to collect deuterium so they can grow in an unlimited fashion, and this is how stool is produced. And I'm just mentioning this because we are doctors and we are trying to understand this process. Stool is practically dead bacteria and yeast and protozoan and all that it lives in your gut with deuterated substrates that are not used and cannot be used by your body. And this is the same as, and there are many interesting papers describing the role of the microbiome to return propionic acid or a ketone body from lactic acid. Instead of metabolizing glucose or lactic acid and produce glucose in the Cori cycle, which is a liver-bound process, it seems that long-distance runners, they develop a bacteria for that matter, just to produce propionic acid from lactic acid to protect their nanomotors. And this is why they can run 40 some kilometers when they run the marathon. So, and you have to train for that. And the training is nothing else from the metabolism point of view than how to deplete deuterium from your diet and protect your nanomotors so you can do a long distance running and you can use your muscles for that matter. We believe that this control of deuterium and depleting deuterium is the main purpose in biology of male mating fights and mating dances, practically in ketosis, meaning that when animals are in the mating season, they don't eat. The males don't eat, but they fight. And practically it's comparing their nanomotor, their strength, their focus, their ability to fight off competitors. And those will provide the best chance for fertilization. The strongest male has the most efficiently working nanomotors with the least deuterium in their system. And this is probably why mating dances are so important, because animals, they can smell and they can taste deuterium in liquids, water, and body fluids. Humans, we were able to smell and taste deuterium in caveman times. And actually, there are studies that show that our taste buds or our smell receptors or receptor cells that communicate with our brain, they generate a different action potential based on the deuterium content of the water, for example, that gets in contact with the mucosal membranes of the mouth. Deuterium-loaded water seems more sweet, or we were able to taste the deuterium content of water back in history. And when we look at anthropology, we can still show that our taste buds respond to the deuterium content of the food and the liquids that we consume. And I think we need to learn again, and this is, I would assume, would be a very important topic to cover for medical students and for doctors, that we may need to again learn how to taste, how to select low deuterium food and low deuterium drinks, and how to control any chemical that get in our body through their deuterium content, meaning that I usually recommend just drink as much as is necessary to clear your thirst after you waited a few minutes if your thirst still bothering you. And this is the best way to produce antidiuretic hormone, or ADH, that actually returns your more valuable metabolic water that is produced in your body if you consume enough fat to produce water. Because 1 kilogram of water, 1 kilogram of bacon, for example, will produce 1.1 liter of water in your mitochondria. And if this is carbohydrate, it's only half a liter. So half, as a little bit less than half of what fat would produce. And so is the energy level. Fat produces much more energy simply because ATP is produced by these rotating nanomotors, and fat produce 2.5 times more energy by weight than carbohydrates would or proteins would. So for that matter, this is all almost like mathematics or engine design, for that matter, how you design a sports car with high performance engine and appropriate shock absorbers and your braking systems and your fuel filters, your fuel pumps. It's pretty much the same idea, when you look at the creator's plan to design a living system that is able to operate with a certain input, for that matter, or consuming the appropriate fuel and consuming the appropriate gas to produce energy, for that matter, and to be able to carry on certain life functions without disease and terminal conditions or debilitating conditions like these chronic diseases that you and I mentioned. So, for that matter, I think this is a very fundamental basic knowledge, nutrinomics, that can be used in all aspects of medicine, in my view.
So it's giving the answer to the damage that's really happening at the nanomotor level within the electron transport chain, essentially, which is the creator of the ATP and keeps the ion differences between the membranes and keeps us alive, essentially. And it sounds like our recommendations over the last, at least over the last 100 years, if not longer, to eat a mostly plant-based diet and very little animal fat over certainly the last 50 years. But even to drink too much water, that it sounds like we're not eating fat, so we're not making the amount of water we should be making in our system. And so that we're trying to make it up by drinking water. But is that water we're drinking different than the water we're making in our from fat metabolism?
It is very different. If you burn fat in your body, you produce 115 ppm water, metabolic water. That means each million hydrogens would have 115 deuteriums. If you drink water, potable water from the faucet or from a plastic bottle, from whatever source there is, they contain anywhere from 145 to 150 or even higher amounts of deuterium. So it's very risky to drink water and not to produce your own water from the appropriate food. Again, if you don't eat the appropriate food, then you may produce high deuterium water in your system. And this is the difference between a cow steak that comes from grass-fed animals or a cow steak that comes from grain-fed animals, because their deuterium content, we are actually carrying out studies to measure and compare these differences. But for example, milk fat has 115 ppm in ghee, in natural pasture-fed animals, and if they are grain-fed or farm-raised animals, their deuterium content is 140 ppm. So there's a significant difference of deuterium content of what type of food we eat, what's the source. This is what I was referring to in the beginning of our conversation, that the source is very important, the amount is very important, and the thirst is very important. And thirst is one of the basic body functions and signal systems that we know we need something from the environment. It's like sleep, it's like hunger, and you don't eat a hamburger just because you walk by a McDonald's. For you have to be hungry for that. I would not recommend that, but that's my choice. And you don't sleep just because you walk by a couch or a sofa bed. And you don't drink just because somebody told you just to have a glass of water. You drink when you are thirsty, and you only drink as much as drink when you thirst. I'm not saying don't drink. Of course, you have to drink if you are thirsty and your thirst is not going away. You cannot produce water sufficiently internally because you're out in the sun, you are doing some work, you are, using up your metabolic water because you sweat, and so and then you have to drink. But be careful what you drink. The most I would say appropriate type of drink is deuterium depleted water. There are many sources. I usually prefer water below 130, 125 ppm. Uh, my goal is to be in the range of burning fat, which is good rainwater at higher altitudes, let's say 110 ppm, and eat natural food or fatty carnivore diet simply because that has the lowest amount of deuterium, and you have a chance to balance your water producing system with your energy producing system to expose your nanomotors to the least amount of deuterium that can get into from the environment. And practically, when you look at your nanomotors, they operate like car engines. There's an electric spark, which is the electron transport chain. There's a highly charged proton. Without its electron, it's a positively charged proton that will occupy a binding site on this nanomotor. And this whole force is driven into the mitochondrial matrix simply because oxygen is waiting there to form water. So maintain this gradient because the protons are disappearing. So it's about 250 protons outside of the intermitochondrial membrane, 250 protons compared to one proton that is free at any time in the matrix because water is formed very quickly. It's getting consumed up by oxygen. So that maintains this gradient that will drive these nanomotors to produce ATP. And there are a number of other things that can happen to protons in the mitochondrial matrix, which I'm not going to detail here. I'm not going to go into further details regarding those processes, but just keep in mind that the Ferrari or the Formula One race depend on many parts that actually can serve the power delivery process with the appropriate control at the most optimal range and or manner. And this is what biology is about, and this is what deuteronomics is about. It's practically to be able to control and serve our nanomotors with the highest octane of hydrocarbons that are coming through food and the cleanest oxygen for that matter.
A pit crew. That's you need the Formula One's probably the best one because you need the pit crew. That car can go so much. You got to refuel it, but then you have to change the tires, and I'm sure there may be other things they're putting into that. So plants, which are a ton of sugar, fructose, glucose, and others, lactose, mannose, they provide a lot of deuterium, more deuterium than comes from the fat of the animal. And I'm wondering if the protein tends to contain more deuterium than the fat of the animal in general, because we talk about EC, because fat, I've always thought as fat is the pure fuel for the mitochondria. If you eat carbohydrates and protein, your liver can make the fat, which turns out to be the fuel for the mitochondria, because acetyl-CoA from a fatty acid right in the cytoplasm of that cell, you can make it so fast. It doesn't need to take glucose to pyruvate and then make acetyl-CoA. So it sounds like if you're eating a high plant-based and a low animal fat diet, you essentially have a high deuterium exposure.
Low, yeah, you're very correct. It's practically just if you look at your airplane before you take off, you take a little sample of the fuel. You have to check if it has any kind of contaminants, water, sand, whatever. You call the tower and you cancel your flight plan. And this is the same with food, practically. And you're very correct. When you eat plants with higher deuterium, with higher sugar content, with higher cellulose content, with a higher fat content that is higher in deuterium, then fat can actually shoot into your mitochondria directly through the carnitine transport system, and it can be oxidized. If we are talking about palmitate, which is a 16 carbon fatty acid, it's going to give you 8 acetyl coenzyme A units and acetates, and you harvest 2 protons from it. And if this fat is not the appropriate fat, there is no filter system. Just like glycolysis has 10 steps, and glycolysis has 10 steps to make sure that the glucose that you eat is, all the hydrogens on glucose are replaced by low deuterium metabolic water hydrogen that is produced in your mitochondria. And I have delivered these talks at UCLA, and you can find these talks on my YouTube channel, where actually we can discuss in detail, or I am discussing in detail what you have mentioned. And this is a very important, very critical point of understanding the system, and that is you have to be careful with fat, because fat or acetate units, they get into your mitochondria through a protein receptor system. There's no proton exchange or water exchange in your plasma that carbohydrates have to go through. And if your body is not low in deuterium, or your cellular water is not low in deuterium, then you cannot really replace all the protons or the deuterons or deuterium in your fructose, carbohydrates, sugars, proteins, or amino acids that come in, which are high in deuterium. You cannot replace them with protons to protect these nanomotors, because eventually all fat is produced through citrate synthase. Now, citrate synthase is very important. It's a mitochondrial enzyme. There's no citrate synthase outside of your mitochondria. And because citrate synthase is a mitochondria-based enzyme, that's why it always uses deuterium-depleted mitochondrial matrix water. Citrate synthase, or citrate, is the source of all fat in nature. Because citrate gets shuttled out, it's called a citrate shuttle. It's getting shuttled out to the cytoplasm, and the cytoplasm has this fatty acid synthase complex that uses this citrate product. It's called melanin coenzyme A, but practically those are the building blocks. Citrate is the source of fat in all living systems, and it's a mitochondrial process, and mitochondria in nature uses deuterium depleted water, so all fat is deuterium depleted. To what extent, it depends on your feeding protocols, your nutritional, it depends on many factors, the environmental factors and so on, but if everything is handled in its natural context, meaning that the animals are grass-fed, pasture-fed, there's no high carbohydrate containing corn, or supplements from jellyfish, or Halloween candies, leftover Halloween candies that they grind into the animal feed, then your cows are able to produce from grass, Book of Genesis,
Their historic diet forever, right?
Practically every living creature, I give them green grass fat or green grass as in the bottom of the food chain. That means every source of meat and fat has to come from a natural source in nature, and that is low in deuterium as much as necessary for mitochondrial processes, and all fat that is produced in the animals or all hydrocarbons. And I'm saying these hydrocarbons simply because they are not carbohydrates. There's Hydrocarbons have a much more and higher ratio of carbon hydrogen than carbohydrates. They mostly have hydrogen attached to carbons. Carbohydrates have hydrogen, oxygen, and hydrogen uh, attached to carbons, but it's kind of mingled up with oxygen that is really not necessary for mitochondrial functions because it comes in through oxygen from air. And for that matter, this is very critical. What you mentioned here and what we covered here is that this mitochondria is practically the essence of creation for that matter. Nanomotors have moving protein parts that have no imprints or have no versions in evolution. It's practically, when you compare this whole system to the industrial evolution or revolution, you had to go through steam engines, then internal combustion engines, then diesel engines, then electric engines, and so on. So the power source have changed in industrial revolution. It's not the case in biology or biological adaptation. Your power source, your nanomotors are very much the same as they have been ever since there's life. So there are no other prototypes of nanomotors. And some of those proteins did not and could not evolve gradually because they would not have any role in any other part of biology other than being a rotating protein and a nanomotor. So this is a very important key element of understanding the importance of the carnivore lifestyle using grass-fed meat source, mostly fatty steaks, eat once a day, go on like, you know, certain religious,
Fasts.
practices. You just don't eat for days. You just let those fat sources, those acetate units, be part of your water, uh, matrix water and mitochondrial water and metabolic water to keep your body and you keep your mind clean and to be able to um, have a healthy body function that includes practically almost everything that is concerned as far as depleting deuterium through food and dieting and lifestyle.
So the science of deuterium has really taught us that there is the electron proton, looking at the atom and the electrical charges that are going on are ultimately have the damaging factors to the nanomotor, which ultimately the mitochondria dies, the cell dies, enough cells die, then the organism dies. And so fasting is a very important component, one meal a day, and whatever those time frames are, whether it's every 30 days or every 60 days or 90 days, I don't know that we know that one. I like the one meal a day as one of the very best to keep your damaging sugars or maybe even high protein, which may have a lot of deuterium in it. But ultimately, the animals that are fed regenerative, grass-fed, free range probably have the lowest deuterium levels. That's what you're saying versus modern industrial farming, which has a lot of grains and GMO foods, I imagine, and maybe fertilizers that are not natural, that are all contributing to a higher deuterium, heavy hydrogen level, which damages the Formula One motor or the Ferrari.
That's very correct, and this is why I like your talk, simply because everything you say, and it's not a like an overstatement, but everything you say and all the investigators that you quote and other people you talk to, you address deuteronomics one way or another. And everything you say makes a lot of sense from the viewpoint of deuteronomics. Because indeed, this is the nutritional, and as far as lifestyles matter, this is what the appropriate approach is to protect those nanomotors and to protect the human Ferrari. Those are very high-end, high-performing machines. They are more like Formula One race cars. Some of those biological functions require just extremely powerful power sources, nanomotors for that sense. But if you look at a Ferrari, they are designed to perform in an exceptional manner, but all parts have to be protected and designed to this power source. And it was, I think Jay Leno was showing one of the Bugattis with like 1,200 horsepower, and they had to design a brake system, they had to design a crankshaft, they practically had to design all parts based on the performance of the motor or the engine. And this is what mitochondria has to do, or this is what mitochondria have to do to design all our cells have to be the utmost design is what needs to be followed when we operate these nanomotors. And I have to give you a great compliment as what you talk about and what you recommend makes a lot of sense from the medicinal biochemistry and from the deuteronomics point of view, and I have to thank you for that.
So, though I really appreciate that, Laszlo. I came to this deuteronomics is sort of like, whoa, this is really kind of scary stuff. But it really is, as we talk about the nanomotors and, leaded gas versus lead-free gas, or the premium versus regular, it kind of makes a little more sense. I'm trying to understand the nanomotors, and that might be a whole nother discussion, to just understand the nanomotors and how critical they are. Are they also in other parts of our body besides the mitochondria? Or is it really that single moving protein component that is responsible for essentially maintaining the battery, essentially?
This is a very good question. They are everywhere. They are everywhere, but they rotate in different directions.
Okay.
Only mitochondria, because of the proton gradient, they can produce ATP. In every other body cell part, like cell membranes, ion pumps, they consume ATP and they rotate the opposite direction, and they move proteins that pump nitrogen, for example, or chloride, or this is how we maintain our ion gradients.
Mm-hmm.
This is how we operate our cells at the mucosal layer. This is how we kind of maintain our homeostasis, simply because there are nanomotors in the mitochondria that can produce ATP using fat, burning fat in the presence of oxygen, and the product is carbon dioxide and water. And this is what our eye is doing. Practically, the melanin layer in our eyes are able to, in the retina, are able to break water into hydrogen gas and oxygen gas that can actually operate nanomotors in deeper cell layers where blood does not get into, simply because those are metabolically sequestrated from blood circulation. And this is what our pumps, ion pumps do, and this is what our V-type nanomotors do. They actually produce they are called V-type nanomotors. I just reviewed a paper for Scientific Reports Nature where they actually describe these V-type nanomotors that produce these proton-filled gas tanks that can fuse with mitochondrial membranes and supply more protons for these nanomotors functioning in the mitochondria. And they all produce hydrogen gas or pump protons into these vacuoles. That's why they are called ATPAs, because they are ATP. They actually use ATP. They break ATP to pump protons into these vesicles that can actually fuse. It's like more like when you put extra gas tanks on your airplane because you're flying from Syracuse to Los Angeles, and your aviation control requires certain preparations for that, and you have to use extra gas tanks.
Or a supercharged engine, I guess.
Or
even
supercharged engine, for that matter.
Yeah.
And then you're gonna check your gasoline. I guarantee you that, because simply you don't take the risk of ruining those high performing engines. And when you look at your plate when you eat, and this is what I tell my students, listen, you have to think of your nanomotors. You have to think of these rotating little proteins that, from each, in each mitochondria, there are 320,000 of them. And each cell has an average of 1,000 mitochondria, and you have some 30 trillion cells. I mean, you can imagine how many nanomotors we operate in the same time in our body. And when you look at your plate, when you look at your drink, when you look at your, like when you eat or how you satisfy your hunger, you have to think of your nanomotor. That's what you think when you pull into a gas station. You're not going to put diesel fuel in your gasoline engine and vice versa. I saw a funny picture when a guy pulled up in a gas station with a Tesla because he didn't know it's actually an electric car because the car was stolen. So even when you steal a car, you have to know what's your next refill going to be. So it's a risky business when you eat and drink stuff, because you're actually protecting or ruining your nanomotors for them.
So alcohol, where does alcohol fall into? It likely is high in deuterium. It's alcohol, sugars. Any comments on that?
Yeah, so it's a fermented product, alcohol, and depending on what you drink, beer, wine, or spirit, or distilled liquor, deuterium content depends on what the source of that particular alcoholic drink is. We measured wine; it was in the range of 160 ppm, as I remember correctly. But again, that's a plant; it's a fruit-based drink. Beer may be even worse than that. If you drink a bottle of beer, it's more like eating a loaf of bread as far as sugar content and so on. The best is practically distilled liquor if you look at deuterium content. And again, we do have now in the medical literature certain papers and publications. I just wrote a comment to this new study that's out by Dr. Gabor Shomyai, and he actually measured deuterium content in certain food items. There are papers and publications that you can now read, like what to eat, what not to eat, what to feed, what not to feed. We published a number of papers in this scenario. So there's a great deal of information in the peer-reviewed medical literature that you can use for this purpose. And there is now raw data and there's peer review data. Well, some papers go through the peer review process right now. I do comment, and as an editor, I do require authors now to address the deuteronomics part of their papers. We call it deuterpretation, which is the interpretation of their data from the deuterium point of view, and there is a new nomenclature that is coming out with this regard, practically deuteronomics, deuterpretation, there's deuterium accumulation, there's deuterium or proton destabilization, and so on. There are a number of terms, and there's a number of concepts that are entering the medical literature, the peer-reviewed scientific and medical literature. I do like, as a reviewer, as an editor, to address when it comes to mitochondrial functions, when it comes to dietary interventions, when it comes to nanomotor functions. I do require now authors to address the deuteronomics aspect of their work. We do have a number of lecturers and students who attend these courses. Submolecular medicine at Bry University, about 30 lectures cover these topics, just how to use deuteronomics for health and strength and mental cognitive abilities and maintain those functions at the optimum level. I think deuteronomics is going to be very important and critical part of all aspects of medicine, including reproductive medicine, which anytime when you see it's appropriate, I'm very happy to cover those. We do deliver talks at workshops addressing deuterium after all, but it's practically your area when you talk about diets and so on.
Well, this is such an important conversation, and I'm just fascinated to learn so much about something that I was like, oh gosh, it just sounds so hard to understand this, but ultimately it's such an important thing. Deuterium, which it sounds like it goes along with a lot of the damaging diseases that you've commented on, hypertension, diabetes, cancer, stroke, and everything. Ultimately, the damage, it comes from our nutrition, which comes from the environment, how we're all sort of misinterpreting how it's all happening. And by going keto, carnivore, fasting, it's really, really helpful here. And I was thinking about how I've been studying a lot about glycobiology, the glycobiome, and by damaging the glycobiome, and it sounds like it's goes along with the deuteronomics, and I'll get that word straight eventually, but I think we're going to have a follow-up on this conversation, Laszlo, because this is so intriguing. Do people, just one or two more quick questions, and we've gone over a little bit, if that's okay, a couple more minutes. Would that be all right?
Yeah, fine.
Good. Awesome. So for those people, is it, we're checking hemoglobin A1C, we're checking blood pressure, so many things we test to look for evidence of why people have diseased and are damaged. Is measuring a deuterium something that's clinically relevant? Are people able to do that? Is it, what's your thoughts on that?
Yeah, this is a very appropriate question. There is ways of measuring deuterium from saliva, urine, and breath. The breath samples are the most revealing of your metabolic water content. In an optimal scenario, it should be below 130 ppm. Usually what we measure is above 140, 145 ppm, unfortunately. So we can see the deuterium concentrations as they relate to disease scenarios in this whole chronic disease arena of epidemic, significant metabolic diseases appearing at like pandemic rates, practically because of their high prevalence. But as you said, the nanomotor understanding deuteronomics, deuterification, and you know it or not, but you're one of the best deuteronomicists because you apply, just to throw another term, it's just practically that's what you do. You as a physician, you address nutrition from the natural source, how many times in a practical manner from experience and from your best clinical judgment simply. And that's what it takes to address in the same time deuterium for that matter. And you do this in unintentionally, and from now on I hope this is protecting your nanomotors and looking at your plate as you look at your gas pump. It's like, I actually, when I pump gas, I actually check it. Okay, that's diesel, so that's fine. Because I'm paranoid about this. And this is how I eat. I just have to look at the stuff that I eat, and I have to spend money on. Go ahead.
Good quality meats. And we willy-nilly put anything and everything in our mouths. And that is deteriorating us to too much deuterium.
That's fantastic.
And that is really, and we're going to be talking more about the nanomotors and how important it is to keep your deuterium levels below 130. Was that the number that you used?
Well, in breath, when you measure, you want to be at that range, but in your food, it's probably better in the 120, below 120 range. And because you're exposed to deuterium through vapor, you're exposed to deuterium through food, through your drinks, and obviously you like it or not, you eat carbohydrates because of the food that comes from stores, even though you're not like educated or not practicing deuteronomics. You are exposed to deuteronomics, and your best judgment, your best scenario is practically to learn about it, to keep your nanomotors running in your human ferry in the optimum condition.
It's important to understand the science, and the science is giving us the answers to how we can live better, reproduce better. And reproduction is creativity, and it's our families, and our future is so important. So Dr. Boros, and we're going to put your link to www.laslogboros.com. People find more about you. And I think in the next month or two, I would love to talk more about the nano motors and more, and how important these ideas are. So anything that I missed that you wanted to at least make sure that our community gets a chance to hear?
Just please keep going, and if you have questions, just let us know, and we're happy to help you out.
And you said that you have a YouTube channel that talks about other scientific ideas related to this, and so I want to just we'll give some direction to our community how to get there, go there. And this is really, again, deuterium and deuteronomics. It's a new one for me. I've heard about it. Actually, my community recommended that I get a chance to talk with you and interview you. So I'm really grateful. And from all around the world, we're just grateful to everyone sharing and listening. So Dr. Boros, thank you. Laszlo, God bless.
Thank you, Rob, and thanks for the opportunity, and wish everybody well.
We'll get you up in the airplane sometime again.
Thank you.
All right.